All right, welcome, ladies and gentlemen. Today we are going to start chapter 19, which is going to go into just the basics of viruses. So, first off, let's sort of just start off by defining what a virus is. So, a virus is a very small infectious particle, and it contains nucleic acids enclosed in a protein coat. At its most basic form, it's genetic material and protein. That's it. Um, there are also a class of viruses that also can have a membranous envelope around that protein coat, but every virus contains genetic material and protein, and then some of them have that additional layer outside that protein coat. Now, keep in mind, viruses aren't cells. They don't contain a nucleus, but unlike a prokaryotic cell that also does not do that, they don't have ribosomes, they don't have the ability to do metabolism, they don't have the ability to reproduce on their own. So they are very much what we would consider obligate parasites above and beyond the fact that they just need to be in a host to survive. They need their host for quite literally everything. Um, the other interesting thing about viruses are the way that their genomes are set up. So when we typically teach you as underclassmen about DNA and RNA, our two major genetic material molecules, we talk about DNA being double-stranded, RNA being single-stranded. And yet, when we look at viral DNA or viral RNA, that isn't always true. So their genomes can consist of double-stranded DNA, which is what most kids would typically associate the structure of DNA being, but they could also contain just single-stranded DNA. Conversely, they can contain double-stranded RNA, which most kids wouldn't think about structurally, or they can have that traditional single-stranded RNA. So we classify viruses in a lot of different ways, one of which is their genetic material. So is it a double-stranded DNA virus? Is it a single-stranded DNA virus? Is it a double-stranded RNA virus? Or is it a single-stranded RNA virus? And then we can start looking at things like the way that their protein coat is shaped and the way that they put them together, and then what organisms do they infect, so on and so forth. So their genome is DNA or RNA, and then we can talk double or single-stranded. When we get out to their protein coat, there's also a lot of variation here as well. So that protein shell is what we call their capsid. And that capsid's job really is mostly to be the protective coat for that genetic material. Also, then it can have certain appendages and things like that to help it get into the host and the host cells that they infect. So capsid is just a protein shell, and the way that those protein subunits are built, put together, what we call these capsomeres, then gives all of these various structures that we see. So here is more of a rod-shaped capsomere protein coat from a tobacco mosaic virus. This one has a different shape here. Some of them are more these rods, and then here's that membranous envelope potential on the outside of that capsomere, something like the flu virus. These guys are probably what we study the most and understand the most at this point, and those are your bacteriophages, because they're viruses that actually go after bacteria, which makes it much more convenient to study, because we can grow a bunch of bacteria in a petri dish and be able to study the virus effects on those cells. We also don't have to worry about them infecting us as humans either, which is helpful. So some of them, it's just that capsid, and then some of them can have that membranous envelope that goes above and beyond as an extra layer of protection and an extra bit to help them infect their hosts as well. Interesting thing about those viral envelopes, while we talk about those for a second, they are often derived from the host's cell membrane. So not only do they contain viral molecules in that membrane's material, but it will also end up containing some of the host cell molecules as well, which really makes it an interesting evolutionary progression, and an easy way for that virus then to continue infecting its host's cells because it literally contains materials from its own host. And so that host's immune system has a harder time recognizing that it's present because it thinks it's part of itself, which is kind of clever, if you will, from an evolutionary standpoint. So I mentioned a moment ago, right, bacteriophages are probably the ones that we understand the most about simply because they're the easiest for us to study. We've been studying them for the longest, um, and we can do it pretty easily in a lab setting without worrying about the risk of infection to humans. So bacteriophages, these are, again, these bacterias that are, excuse me, these viruses that infect bacterial cells. You may also come across it as just a phage, same idea. And so we use these guys as a study model for studying viruses in general, just the way that they infect, the way that they mutate, the way that they evolve, structure, pretty much the whole 9 yards. This is where we go to first, and then we start extrapolating out um, when we look at animal viruses or plant viruses that don't seem to quite fit this model, what's happening that ends up being different. So this is sort of our baseline for studying viruses in general.